Well, good morning, everyone. Thanks for coming. My name is Fernando Valbuena. I am a senior studying biomedical sciences at the University of Central Florida. And this summer, I worked with Dr. Andrew Schuerger in a project titled Aerobiology of Atmospheric Dust over Florida, a possible source of contamination on planetary spacecraft at KSC. The objective of this project is to characterize the microbial population present in dust plumes of African dust storms that cross the Atlantic Ocean over to North America during the summer months. Dr. Schuerger aims to test these storms for the presence of possible potential human, animal, and plant pathogens, as well as possible sources of contamination that could affect a spacecraft assembly at KSC. In order to sample the storms, Dr. Schuerger engineered a special air sampling system that you see over here called DART that I will talk about shortly. Once the storms have been sampled, the samples will be analyzed using culture-based techniques and DNA sequencing. This is a picture of DART, which stands for Dust at Altitude Recovery Technology. In essence, it consists of a pump which draws air through a series of scoops located at the front of the cylindrical body. You can't really see it, but it's in this area. Each scoop is individually connected to a hose and a filter holder that contains a filter. The pump draws air through the scoops. The air passes through the hose and the filter, depositing microbes, spores, aerosols on the filter. Then it travels through an extra section of hose and a solenoid which controls the airflow, and finally exits the device through a manifold. DART has been designed to be mounted on the wing of two different types of aircraft. So it's been already flown on an F-104 jet, which you see right here. And for our subsequent science missions, it will be mounted on the wing of a Texan 6 aircraft, which you see here on the left. This is actually Dr. Schuerger flying the T-6. The project can be divided into three main phases. The first one being filter optimization, which is the one we spent most of our summertime on, working on finding a filter material in a pore size that worked best for collecting our samples. Two other important steps of this optimization phase was first to find an optimal growth medium to grow our trapped microbes, and also to select a time of collection for the sampling times. The second phase is the actual sampling of the dust storms that are scheduled for the end of August. DART will be flown at different altitudes to recover the samples, and then those samples will be characterized during the third phase, employing DNA assays and culture techniques. At the end of the project, Dr. Schuerger will be able to determine if there are potential pathogens being carried over from Africa to North America, and he'll be able to determine if the increases in biomass actually pose a contamination concern for spacecraft assembly at KSC. This is one of our earlier data sets for the filter optimization phase. It shows a comparison between two different types of filters, nylon and polycarbonate, of different pore sizes with relationship to airflow. As you can see, there is an increasing trend in airflow as pore size increases, which is seen in both types of filters. But what's interesting is that the trend itself differs depending on the type of filter. So filters with the same pore size will actually display different airflows depending on the material and the design. This was very important for us to understand at the very beginning phase, because we were trying to find a filter with a high airflow, but with a pore size small enough to actually trap the microbes instead of just letting them flow through. This is a second, more advanced data set that we collected during a time study performed over 2.5 weeks. We ran a series of nylon filters of different pore sizes using DART, the back patio of the SLSL facility. We ran each filter for 60 minutes, a total of 4 times, in a completely randomized fashion. The filters were then plated on R2AT, which is a standard growth medium, which contains turgitol in it, a surfactant agent which slows down the fungal growth. We want this because we don't want the fungal colonies to overgrow the plates and not let other microbes grow. The plates were incubated at 27 degrees Celsius for 2 to 4 days. After this time period, they were screened for viable colonies, and the numbers were recorded. As you can see, this is the line for the bacterial cells recovered. This is the line for fungi recovered. These error bars represent variance seen in repeated samples, which means that the numbers of microbes we obtain at each repetition of the filter actually varied. We didn't have a constant number, most likely due to differences in weather and temperature between the different collection days. Although there was not a significant change in the number of bacterial cells recovered across the entire range of pore sizes, there is a clear increasing trend in the number of fungal colonies obtained as a function of increasing pore size. So even though at the beginning we thought that a smaller pore size would actually yield higher number of microbes, this time study actually revealed to us that airflow is more important for the collection of microbes than pore size alone. Basically, more air flowing through the filter overcompensates for the microbes that might be lost as they travel directly through the bigger pores. This is what our filters will look like after the incubation period. As you can see, there are a significant amount of fungal colonies of different morphologies, not that many bacteria. At this point, we would divide the filter into quadrants, count the viable colonies, and record the numbers. The second phase is the sampling of the storms. We have science missions scheduled by the end of August or beginning of September. By then, we'll have selected a filter type and time of collection. However, it's important to note that our experiments were carried on the ground, and variations in air density will inevitably affect the times of collection as we increase altitude. Dr. Schuerger will most likely utilize 2 filters per sample run, one for culturing and one for DNA or metagenomic studies. In preparation for the third phase, we worked on optimizing a DNA extraction protocol from fungal mycelium using samples previously collected during DART test runs. We extracted DNA from the samples after subculturing them. We performed amplification of the 18S region via PCR. We cleaned the product, confirmed the presence of the product, cleaned the product, and sent it up for sequencing. We just got the sequences back, and we will go ahead, work on a contake, and then compare those sequences to those stored in the National Center for Biotechnology Information website to find out the taxonomy information we need to characterize those fungi. Prior to DNA extraction, however, we had to extensively subculture our fungi in order to get them in pure culture, that is a single fungus per plate, as you see here. At this point, we'd see that there's a single type of morphology. We would go ahead and extract DNA from the fungal mycelium, perform PCR amplification, run those PCR reactions on an agarose gel, confirm the presence of the product that you see here, clean that PCR, send it up for sequencing, and then analyze the sequences that we get back. Once DART actually samples the dust storms, this is the procedures that we'll utilize to analyze those samples. The data I generated during the summer actually earned me my first co-authorship in an abstract that Dr. Schuerger will be presenting at the American Geophysical Union Conference in San Francisco later this fall. These are some of our main conclusions. Perhaps the most important being that a filter with a higher airflow works better at collecting microbes than a small pore filter with a low airflow. A 30 to 60 minute collection time seems to work best, but like I said, the air density variations have to be taken into account. Variations in temperature, wind, and the presence of clouds actually seem to affect the number of microbes collected per filter run. The number of fungal microorganisms able to grow on the medium always exceeded that of bacteria. Thus, metagenomic studies will be essential to characterize the entire range of microbes present in the African dust pools. Finally, I'd like to give special thanks to Dr. Schuerger for his excellent mentorship this summer. I'd also like to give special thanks to Space Florida and the NASA Florida Space Grant Consortium. Thanks a lot. Yes?
So when you were doing your graph with the fungi and the bacteria, so normally when we're filtering bacteria, we use a pore size of like 0.22 microns.
Mm-hmm.
So did you do any controls where you spiked it with like a known bacterium to make sure that you can recover, so that that you spike it with the airflow so that you have a known quantity in there so you know how much you might be recovering?
Have an experiment with known quantities of bacteria to see how much DNA we can extract from the filters, which is the main problem we've been having with is very low yields of DNA extracted directly from the filters. So we will use that to see what we can improve in our procedure or what we're doing wrong that is producing low yields. But we did not use that sort of control for the actual culturable experiments.
Did you even just put the filter, like stain it with like a dye and then put it under?
We used unused sterilized filters at the beginning, which corroborated corroborated the absence of microbes on the sterilized filters. That's mainly what we use as controls. But we never plated a given number of spores and then plated that, but that would be a good idea.
Because I'm sure Andy has told you, there's only like 1% of all your microbes are going to even be culturable.
Yeah.
So just like looking at it under a microscope might give you an idea of
All right.
the ballpark.
Sounds good.
Can you go back to the slide on the publication?
Which one part?
The slide on the publication.
Oh.
You're abstract.
Yeah, sure. Okay, so how much of this work is in this publication?
Both of the graphs you see here, plus other airflow data that we're collecting with other types of filters that we didn't include in this presentation because we're just testing them and we need to collect more data before we can actually graph. And protocols like changes that we made between working Andy and I, changed some things in the protocols. Also just overall planning of sampling times, which days to collect, which type of filters to utilize. Yeah.
Andy, is he going to the meeting?
Oh, yeah. If you have the budget for that, I'm glad that.
Can you make a poster presentation?
Well,
is this a poster?
No, I've, and I just didn't have a budget to send it, so I think that you see that.
Give me an estimate of how much it costs.
Well, yeah, I can do that.
This is San Francisco, right?
It's in San Francisco, it's tremendously diverse. But to answer your question about all the data that Fernando helped generate this summer is going to be about 1/3 of the presentation. There'll be about 1/3 about the F-104 flights, and for anybody who doesn't know, we have the F-104 team right here. So I certainly want to thank them for all their support during our F-104 missions. And then there'll be about 1/3 for the new airplane going to. So.
Great. Good job.
Thank you. Any other?
Fernando?
Okay, ready? 1, 2, 3.
Over here for his
Oh, sure.